The last two things to talk about here in chapter 16 relate specifically to eukaryotic chromosomes. So, first of all, for a prokaryotic chromosome that's circular, there are no issues with the DNA replication. You can completely replicate the entire full circle. With eukaryotic chromosomes, we run into difficulties with replicating our ends, and here is why. So up at the top, we've got an example of the end of a linear eukaryotic chromosome. The top blue template strand is going to give the instructions on how to create the leading strand. There are no problems here. There's a primer somewhere upstream, DNA polymerase latches on and continues replicating until it essentially falls off the end of the chromosome and the full thing will be replicated. Where we run into difficulty is with the lagging strand. Remember that the lagging strand is created in those Okazaki fragment chunks. And so what you can see here, multiple Okazaki fragment chunks have been already strung together, and we're still just finishing up here the last two at the very end of this linear chromosome. We're going to zoom in here in this box and take a look at how the cell goes through finalizing the last steps of DNA replication here at the end. So here's your next to last Okazaki fragment. This RNA primer needs to be removed and replaced with DNA nucleotides. No problem. There's a nice free 3' end here in blue, that's DNA, for the DNA polymerase to latch onto and fill in the gap here. DNA ligase will seal the backbone, and now you've got a nice continuous piece of DNA here. We have to do that with this last RNA primer. So that RNA primer has to be removed. Once it's removed, as shown here, we would like to fill in this little end. Trouble is, though, that you'll notice there's no free 3' end on the top strand here for DNA polymerase to latch onto and fill in this section, and so it can't. And nothing is done with the end of this chromosome. So now we have a little section here of this single-stranded DNA that's unprotected. Remember that single-stranded DNA in any cell is highly subjected to degradation. And so there are enzymes that are going to come over and they're literally like a little Pac-Man going to chew off the end of this chromosome, make it shorter until it gets to that double-stranded DNA, and then the double-stranded DNA gets left alone. And so now this chromosome is a little bit shorter than what it used to be. And so initially that's not a big deal, but if you think about those cells that divide a lot, every time they divide, they replicate their chromosomes, and every time their chromosomes get a little bit shorter and shorter and shorter. Well, if you lose DNA, you're potentially losing the genetic information you need to be able to function as a cell. So this is a huge problem, because if you lose too much of that DNA, you stop functioning. So how do eukaryotic cells get around this problem? Well, eukaryotic chromosomes have certain chunks of DNA at the end of the chromosomes that are called telomeres. Now, telomeres, they don't code for anything. Their main purpose is to be lost. They don't delay the shortening of the chromosomes. All they do is they kind of create a buffer, a region of DNA that can be lost with every DNA replication cycle that really doesn't matter to the cell. Right? And so in this particular figure or picture that we have here, these are chromosomes. They're duplicated. So you see the sister chromatids attached to each other. The DNA in general is highlighted in blue. And then you'll see that at each end of each sister chromatid, you've got a yellow fluorescent probe that is highlighting the region where the telomeres are, right? So telomeres are these non-coding regions of DNA at the ends of eukaryotic chromosomes that are meant to be lost because of this issue with the copying of the ends of eukaryotic chromosomes. And some scientists have proposed that maybe the shortening of telomeres is somehow connected to aging, right? The older we get, the more our cells have replicated, the shorter our telomeres are. Maybe that has something to do with the process of aging in our cells and in our multicellular bodies. This is an active area of research in science. One more thing to think about though, right? So we replicate our DNA before mitosis and before meiosis, or the production of our gametes. And so if you think about it, we should, with every generation, be passing on shorter and shorter chromosomes to our offspring, right? Because we have to replicate the DNA before we can make egg or sperm. With every DNA replication, our chromosomes get shorter. So you would expect that the human race would progressively have shorter and shorter chromosomes with each successive generation. This is not the case, though. This is because those cells that give rise to our gametes have activated a gene that all cells in our genome or in our bodies have, and this gene is called telomerase. So we have it in every single cell, but it's only activated in those cells that give rise to our gametes. And the purpose of this gene or this protein or this enzyme called telomerase is simply to lengthen telomeres before the chromosomes are sorted into gametes, so that when we do pass on our chromosomes to the next generation, we literally aren't giving them, the shortest stick in the bunch, right? We are giving them full-length chromosomes that they can then spend their lifetimes shortening. Now, it's probably a great thing that our cells, the rest of them that don't give rise to gametes, don't turn on this telomerase, because it helps to protect against cancer, right? Because for cancer cells, they divide much more frequently than even, the most highly proliferative cells in our bodies. And so if cancer cells divide so much so that they lose their telomeres and start losing important genetic information, those cells are going to die. And that's good, right? We want cancer cells to die. The tricky thing is that cancer cells are sneaky. And some cancer cells have found this telomerase gene in their genomes and they've turned it on. And so these cancer cells now express this enzyme that can lengthen the telomeres at the end of the chromosomes, and they can essentially proliferate forever. Very, very sneaky. All right, and then lastly, let's review a little bit bacterial versus eukaryotic chromosomes and talk about some special things in regards to our eukaryotic DNA. So remember that a bacterial chromosome is still double-stranded. It's still made out of a double helix, but it's a circular DNA molecule, so it has no beginning or end. And in a bacterial cell, there isn't as much DNA. It's usually just that one fairly small circular chromosome. And that chromosome is usually just super coiled really tightly in a region that's called the nucleoid. So no nucleus here in the bacterial cells. For eukaryotes, again, a little bit different. We still have the double helix for our DNA, but our DNA molecules are linear, so they have two ends, right? We just talked about the difficulties with replicating those ends. And then on top of that, eukaryotic DNA is very long. And so the cells have to make sure that they can organize these linear chromosomes in such a way that they don't get tangled and broken and are still accessible for enzymes to go in and read the information that's stored in those DNA molecules. And so eukaryotic DNA is going to be organized using protein spools called histones. And the DNA is literally wound around, just like a spool, wound around these histone proteins. And so the material that your chromosomes are made out of is really this material that we call chromatin. It's both the DNA plus the protein that's used to organize the DNA. So let's take a closer look. Here we've got a nice outline of how DNA is organized in eukaryotes. So in blue, you see the double helix. Purple are proteins. So these are the histone proteins. They're like a little ball with a little tail on one end. And 8 of these histone proteins come together to form a spool. And one of these spools can hold 2 rounds or 2 winds of the DNA. And there's a little segment of free DNA, another little histone spool, 2 more winds of the DNA, a little bit more free DNA, and so on and so forth. And so all of your DNA is organized using these histone spools. And the spools are then all kind of bunched together to create fibers. Those fibers are then looped together into eventually at some point of the cell's dividing a condensed chromosome. Now, even when a cell is not dividing, there are certain regions of the DNA that are more loosely packaged or some that are more condensed. And so in interphase, when the chromosomes are not officially condensed into those discrete chromosomes that we think of, especially when we think of the process of mitosis or meiosis, in interphase, that more loosely packaged chromatin can exist in really loose state or a slightly more condensed state. And we call these two states euchromatin and heterochromatin. Euchromatin is your more loosely packaged chromatin, and this allows for access of those enzymes that participate in transcription. So this allows for gene expression or the reading of the genes in that euchromatin. On the other hand, heterochromatin is much more highly condensed. If it's more condensed, that means that there's no access to the genetic information in those regions by those enzymes that participate in gene expression. And so there is suppression of gene expression in those heterochromatin regions. Typically in interphase, you'll find heterochromatin in centromeres, so the narrow waist of the chromosome, and also in telomeres. And this makes a lot of sense because honestly, there's not a lot of coding information in the centromeres, and we just learned that telomeres, they don't code for anything. So it's probably a good idea to make sure that those are tightly condensed so that they're really not taking up a lot of space in the nucleus. Now, euchromatin and heterochromatin, these areas in your DNA, they can change. So sometimes you can more loosely package parts of the DNA, and other times you can cause it to condense a little bit more. And you can do this two ways, either through modification of the DNA itself. We talked about this. We talked about how you can methylate or add methyl groups to DNA, and this causes the DNA to condense. You can also add a variety of different chemical groups to histones, and this will encourage the histones either to pack more closely together to create that heterochromatin, or to kind of spread apart a little bit to create the euchromatin as well. And then lastly, we've been able to see how chromosomes are actually organized, not only in cells that are going through mitosis where the chromosomes are condensed, but also in interphase when their chromosomes are more loosely packaged. And to do this, we've used a technique that's called chromosome painting. And so we have devised fluorescent proteins of different colors let's say fluorescent probes of different colors that we can use to bind to specific parts of the DNA, so different chromosomes. And so we can paint chromosomes of different colors. And so here you see a karyotype where we've used this chromosome painting, and this helps to match up the homologous pairs, right? They have similar DNA, which means that the probes of that particular color will bind to both of the homologs in the pair, allowing for easier matching. But we can also use these same fluorescent probes to probe for chromosomes in phase nucleus as well. And so you can see here, it's beautiful, right? You can also see how the chromosomes, they're not really all tangled in one big mess. Each chromosome kind of has its own real estate that it hangs out in within the nucleus.